All right, I'm going to talk about something completely different. I was asked to come here and talk about work that my group has been doing recently with soil crusts. So just in case you have to run to a dentist appointment or something, the answer to my question is yes, we can grow biological soil crusts, um, but we'll take a minute to get there. Um, First, I want to tell you about what we're talking about here. I think in general, people in this room are probably aware of biological soil crusts, but maybe you don't know some of the details about them. I like to introduce them to people as the charismatic microflora of the desert. They include such memorable characters as these colorful lichens, mosses, cyanobacteria, really in any combination. And these grow on the soil surfaces in dry regions of the world. So for sure, they're players in upland systems. Might they have a role in riparian systems? I would say, yeah, probably. Um, Not perhaps the same eco-hydrological niche as cottonwoods and willows, or not in wetlands. But if you look at the whole riparian zone, we also have these upper terraces that flood infrequently, occupied by species like mesquite or maybe greasewood, and that would be very plausible biological crust habitat. So right now we're witnessing a grand experiment where we're seeing a lot of this beetle-affected tamarisk canopy. It remains to be seen how much of this is actually going to die or go away, but perhaps some of it will. And we might be sort of having an opportunity where we've got a lot of riparian and terrace real estate that's suddenly available for something. And whether that something is good or bad might be up to our actions. So, getting back to our crusts, what might crusts be able to do in that might enhance ecological restoration of former tamarisk stands? Okay. So I'm just going to give you the 1 minute version. This could be the subject of my whole talk, and I don't want to do that. But number one on the list always is aggregating soil. Sort of the calling card of this community of organisms is to create a continuous horizontal soil aggregate. And if you have soil aggregates, that means that the soil is much more difficult for erosive forces to move. So this is erosion proofing of soil. This little ledge you see here, that's created by, it's basically wired together with cyanobacterial filaments. You can even see them. They look like little cobwebs. Most of the organisms that dominate these crusts are photosynthetic. So that means you've got not just plants as a pathway for carbon into the system, but you've also got a photosynthetic soil surface. There's another way for carbon to get in. And eventually that carbon is turned into organic matter. Organic matter is very important for long-term maintenance of soil fertility. It's both a nutrient reservoir and it's a colloid. It holds on to nutrient cations. Also living within crusts are several nitrogen fixers. So usually if you have a crust, especially if it's black, you have organisms that are capable of grabbing the world's most limiting plant nutrient, which unfortunately is almost all in the atmosphere, grabbing that out of the atmosphere, pulling it into the soil. Pretty neat trick. And crusts are also important and influential for their hydrological effects. This is actually sort of a controversial thing because crusts do different things in different parts of the world. But in general, it's thought that in North America, because most of our crusts have this bumpy texture, they are actually acting to enhance water capture and promote infiltration rather than the opposite, which would be to shed runoff. So we have sort of a water harvesting system here. Okay, so those are all useful capabilities. Why don't we use these organisms in restoration? I think the answer is simple. It's just because we haven't put in the time and effort to figure out why. We've never gotten past the research and development stage. We've hardly even started it. We're nowhere close to being able to go out and use crusts on a multi-hectare restoration project like we heard about in the last talk and in several talks. Does that mean we can't do it? No, it just means we haven't. So here's how we do it, or here's what we've learned. There are various ways to go about growing biocrusts. Most people have focused on the cyanobacterial component, and the cool thing about them is you can isolate them in a liquid medium using microbiological techniques, and you can grow them up in large quantities. And although not all labs that have tried this have actually succeeded, at least one lab in the world, in China, has been able to get this to go big scale, like big outdoor ponds, raceway ponds, and bioreactors. So they've basically got this cyanobacterial soup, which can then be used as a liquid inoculum, and they've been able to grow crusts at hectare scale pretty rapidly. So that's pretty neat. Like I said, only one lab's had that much success, and we don't know why. We think they have one strain that's really weird or something, but no matter what their secret is, it seems to work. My own research group has been taking a little bit different tact, a different approach. We felt that because lots of people are interested in the cyanobacterial component, perhaps we're ignoring some of the other organisms that exist in biological crusts. And we knew that it is possible to grow mosses. So that's where we put our focus. So we started setting about figuring out how to grow these mosses in different combinations of growing conditions in a greenhouse setting. Now, if you're going to grow something, the first thing you want to know is how does it reproduce? Okay, so bear with me for this small diversion. It's actually kind of fun. So this is a moss stem. This is the most dominant crust moss in North America, Centrichia canonervis. It's about 2 millimeters tall. Okay. Now, when you look at these things, 80% of them are functionally sterile, meaning that they've never expressed their sex. They've never grown sex organs. They're incapable of sex. 80%. Now, of the 20% that are not sterile, there's a real problem. They're all female. They're almost all female. It's a 14-to-1 female to male sex ratio bias. Okay. So the first question you're asking now is, how do you know the sex of a moss? The normal way to do it is you take its clothes off and see what's underneath. So basically, here's a moss stem, all of its leaves stripped off under the microscope. And this one here, see these red appendages? Here's a closer shot of that. That's an archegonium. That tells me that's a female moss species. This is what I did my undergrad research on half my life ago. So this is near and dear to me. So this is a big investment for this little plant, but check this out. This is what the male has to do. Okay. My voice changed, and I got really tall when I went through puberty, but I didn't have to double my body mass growing some outrageous appendage. Okay, so this is probably the reason for the male-female sex ratio bias, that the male needs to have abundant resources just to grow this thing. Okay, so now if that happens, and miracle of miracles, this guy is next to a sexually expressing female, and a fertilization happens. By the way, this is not done with pollen. This is done by swimming sperm. So there has to be contact of a water film between this male plant and this female plant. So if all these improbable events come to pass, then the female has to grow this thing. This is the plant down here, and this stalk is a sporophyte. Okay? You were feeling bad for that male before. But now look at this poor female. This would be like a human growing a street lamp out of their abdomen. It's an outrageous metabolic cost. Okay, so this diversion, all this is about really is the fact that sex can be really challenging for some of these species. And plants that are sexually challenged like this, they tend to be masters at cloning. Okay, and this species is no exception. This species is a master of cloning, and we can use this cloning ability to grow them. So some of the best features of mosses as restoration materials are on this list. The first one is that clonability. And I'm using a fancy word, totipotent clonability. Totipotent means that basically any tissue can grow into a new plant. Yet again, a neat trick. These guys are full of neat tricks. They're also highly desiccation tolerant. That does not mean that they can hang out in dry environments. That means that they can lose all of the water in their tissues and still be alive and rehydrate at a later date. Okay? That is very useful because that means that you can have a viable living propagule in dry storage, potentially for decades. You don't have to grow them on demand. You don't have seed viability issues. You just keep them dry, cool, and dark, and they're viable. When they wet up, they pull in an outrageous amount of water. Okay? This relates to that water capture ability. Some moss species pull in up to 14 times their dry mass. When you have mosses, you don't just have mosses, you also have possible symbionts. Some symbionts are cyanobacteria. They're desirable because they fix nitrogen. They live in between the leaves of the plants. And also, they may harbor mycorrhizal fungi. So we could envision a scenario where if you have a site in need of restoration, you want to deliver mycorrhizal fungi to benefit vascular plants, maybe the mosses can help us get them there. And then finally, these species, high pH is very normal to them. Salinity is very normal to them. Sodicity may pose a problem for many species, but there are sodicity tolerant species as well. One final reason, mosses are often primary colonizers. This is a new angle for our lab. We've been looking at mosses that grow after forest fire. This is a stump hole a few months after the Wallow fire in Arizona. This was very high intensity. The roots burned out there, and you can see this fuzzy carpet of mosses is taken over, sometimes growing directly on charcoal. If you needed more convincing, this, this sock is being colonized, probably in a matter of, I don't know, a few years. It can't be that old. I think it's cotton. So just more evidence that these things can colonize some harsh environments. Okay, how have we been growing them? A grad student of mine and a postdoc of mine, Kyle Doherty and Anita Antoninka, designed this really fun greenhouse ecosystem here. We nicknamed it the Bryotron because we wanted it to sound really scientific. On one hand, all it is a bunch of Tupperwares and tubing. But then on the other hand, like this is a really, really elegant system that uses wicking. So we have a nested basin system. This basin is full of a bed of sand. You sprinkle your field collected inoculum on top. Water comes on when the timer tells it to, fills the lower basin. This basin has wicking holes. Water wicks up, creates a temporary water film on the surface, hydrates the entire surface gently, then water shuts off, everything drains out to field capacity. So you get this perfect watering event right when you programmed it. Not bad for a bunch of Tupperware. So these are some of our results. I'm not going very data heavy today, as you can tell, but we started off our work with 2 late successional desert species, Centrichia cananervis and Centrichia ruralis. We were trying all different kinds of fertilizer regimes and hydration regimes. I don't want to get into those details today. Instead, all I want to show you is that this is where we started, and in our best treatments, we were able to increase that about 6-fold in a few months. So, you know, if you know one thing about crusts, you know that everyone always says how slow they grow in the field. This is the kind of growth that you might see in a decade in the field. but we've been able to prove it's not hardwired in the organisms. You can make them grow. Thanks. Quite accidentally, we also grew lots of cyanobacteria. These are those hitchhiking symbionts we're talking about. Here you have this reddish-brown mat in between some moss stems, and here you've got another one. This is cytonema. This is nostoc. Some people would say, oh no, my experiment's contaminated. We said, sweet. We love that. Here's how it looks like when you're looking at the whole community. This is the same experimental unit. This is at 1 month, and remember, we just added a little bit of moss, like a teaspoon of moss. And we didn't even know there were cyanobacteria in there. And you can see the growth we got over 4 months in this pot. We can reach 100% cover in 5 months. Moss increased a lot, but most of what you're seeing here is the cyanobacteria. So they both grow. One thing that was obvious when we did this was that we were creating a true crust. We were creating a soil aggregate on the surface of these pots. Also, from a functional perspective, you may wonder, if you grew nitrogen-fixing cyanobacteria, did they fix any nitrogen? So we measured this, and we found that it didn't matter if we started off our pots with Centrichia canonervis or ruralis. We did get quite a bit of nitrogen fixation, and it could be modeled based on the amount of cover of those cyanobacterial hitchhikers. Okay, so we can grow mosses, we can grow cyanobacteria. I also told you that lichens are part of these crusts. Can we grow lichens? I'm not aware of any other group that's ever reported any success with this rapid growth of desert lichens. But you can be the first to see these data. And there it is. So again, we started with a little bit of sprinkled lichen material from the field. And you can see what happened after about 5 months. In this case, although there are cyanobacteria growing, most of this is actually lichens. We tried out a few different species, sort of screening their greenhouse growth capability. Here's our two mosses I've already been talking about. We grew them along with lichens. And this is the lichen I just showed you. It's called Kallima tenax. And here's 3 other lichens we worked with. And it was really the Kallima that was the showstopper. It grew much, much faster than the other species. It promotes the greatest total cover of crust. And if I were to a priori pick a species I would like to be able to grow, it's this one, because this is the heavy hitter of the dryland nitrogen cycle in the Colorado Plateau. Okay, so all that stuff, that was just in small Tupperware units. We started thinking about how can we do this bigger. Eventually, you know, once we get our approach dialed down, we might want to devote whole greenhouse benches to this practice and start producing inoculum for outdoor experiments. So that's what we have. We have the same principle, except applied to big flats. So we have whole benches, in this case, devoted to growing Centrichia. And in this case, we're getting very close to a full-on moss lawn. It's been some of our most successful moss growing yet. And you remember the sporophyte, the thing that happens after there's a fertilization? Those are very rare in nature, but we're starting to see them here. So apparently, if you keep these guys hydrated for weeks, then all of a sudden it's sexy time. Of course, we want to take this out into the field. We want to see, can our greenhouse-grown buddies live out in the harsh world? I don't have any data to show you today. I just wanted to let you know we are working on this, and we will have data in about a month. But we're looking at different ways to enhance success of the take of these organisms in the field. Some ideas would be pre-roughening of the soil surface, or taking the edge off with some form of shade provision, or a one-time only watering. But I don't have anything to report today. Okay, thanks for listening. I just wanted to tell you about a couple upcoming projects I end most of my talks like this because since I became a professor, I find that I never do science with my own hands. I'm always fundraising, so I want to let you know about areas we could use funds for. Um, we have a new project going in at MPG Ranch in Montana. Very excited. I want to make this our northern empire of our practice. Also very exciting, near the epicenter of the Tamarack beetle explosion, Dugout Ranch in Utah. We're just going to be getting started on that this spring, and we're hoping to expand that project. And we are also working in Bandelier and Oak Creek Canyon, which just burned in the slide fire last summer. Okay, here's my summary. We can grow biocrusts pretty fast too, in a matter of months, not years. We've grown mosses. This includes desert mosses and post-forest fire mosses. We've grown lichens. We've grown cyanobacteria. These organisms are seldom used or even talked about in restoration, but I think we should start doing so. And in order to get there, I think this is a situation where we need sustained effort on the research and development end to get to a point where we can apply this. So thanks for listening, and I'll take any questions.
Thank you, and good talk. And,
Thanks.
Yeah. I was wondering, would you know there's a species of some kind of crust that grows pretty commonly on sort of first terraces underneath junipers with black grama. Does that ring a bell for you? Because there's actually communities that are very involved with the crust that goes with them.
Mm-hmm. Well, that's not enough to say who exactly is there, but it sounds like most likely you've got probably a cyanobacterial community, maybe peppered with a few mosses and lichens.
Okay, it's very sandy, and so it seems like if you want to see it sometime, I'll show it to you.
Yeah, that sounds cool.
Good stuff. And it's actually a floodplain type of situation. So.
Mm-hmm. Yeah, in sandy soils, usually what happens is you have sort of a platform created by filamentous cyanobacteria, and then on top of that, other organisms come to live once it's stable. And the dark color that you're probably seeing, that comes about due to organisms that are stuck on the surface, and they have to be protected from the sun, and the black is their sunscreens.
Oh,
great. They're loaded with sunscreens.
Awesome, thank you.
Mm-hmm.
My question is, so how do these crusts do in a highly disturbed regime like riparian areas where you get a lot of flood events?
Yeah, you know, if it's an active channel, I can't see that's not a place where you'd expect to see a crust persisting. I would expect to see crusts more on infrequently flooded parts of the terrace, and it's possible that in a flood event that they would get buried and partially compromised, but then recover later over a sort of a decadal sort of cycle.
Thank you. So one of the things that we look at when we're doing restoration is often the reference condition, and so kind of trying to figure out where these different species might grow naturally. And I know personally, looking out on the landscape, I'm always kind of curious areas where I don't see a lot of cryptogam cover versus the ones I do. Mm-hmm. I know there can be lots of different factors involved. I'm just kind of wondering, you mentioned that these are obviously adapted to harsh environments, but perhaps maybe what are some of the characteristics that might promote their growth on the landscape already and places where you would expect to see them? And how do you explain areas where you don't see anything?
Okay. Yeah, not every habitat's created equally. Sort of the parameters are physical and chemical soil properties and the climate and the disturbance history. So you could not have crusts for any of those reasons. First place I'd look is disturbance history. If there was chronic grazing in your area, that could be enough explanation right there. But then with the soils, usually sandy soils are good habitat. So are fine soils, really. But where you run into problems when you have a lot of shrink swell clays that expand when they get wet. And like I said before, salinity is not generally a problem. High pH is not generally a problem, but sodicity could be a problem in certain areas. So if your base cations are really sodium rich rather than calcium or magnesium rich, that could be an issue. And with climate, generally, the wetter you are, the more well-developed your crust can be. That said, you still find crust in some very dry places.